Okay,
so
so our project is the effectiveness of rivastigmine on neurodegenerative diseases. Our advisor is Ms. Ranganathan. This is presented by Finna, me, Reya, Sania, and Anshika. Next slide, please. Okay, Alzheimer's disease is one of the most prominent ailments which Americans aged 65 or older suffer from. Today, approximately 6.5 million older Americans suffer from Alzheimer's. As the number of Americans above the age of 65 increases rapidly over the course of the next 30 years, so will the prominence of Alzheimer's, which is projected to affect 12.7 million people, which is almost double today's number. Alzheimer's can eventually progress into dementia, which induces greater memory loss and cognitive difficulties. Although modern medicine is still quite a ways from finding a cure for Alzheimer's, there are several hypotheses as to what causes it. The cholinergic hypothesis suggests that a deficiency in acetylcholine, the neurotransmitter for memory and learning, leads to cognitive decline. The tau hypothesis explains that an aggregation of tau proteins causes tangles in the brain, leading to cognitive impairment. The amyloid hypothesis suggests Alzheimer's is caused by abnormally folded amyloid beta proteins, leading to plaques forming in the brain, disrupting neuron communication. Today, several types of treatment exist for Alzheimer's, although none of them work as a definitive cure. Cholinesterase inhibitors prevent cholinesterase from breaking down acetylcholine, which is the essential neurotransmitter for memory. Other treatments include the NMDA receptor antagonist, which block NMDA receptors that cause an influx in calcium, causing cells to rupture and die. Overall, Alzheimer's is still a very prevalent disease in today's society and affects many people in our country and will continue to do so in the future, which is why finding a cure is an issue of the highest exigence. Next slide, please.
So as life expectancy increases with improved healthcare, neurodegenerative diseases will likewise become more prevalent in an aging population. Alzheimer's disease, also known as AD, alone is expected to grow to 30 million cases in 2050. Furthermore, it is estimated that by 2050, the global number of dementia cases will nearly triple from 57.4 million to 152.8 million. While Alzheimer's disease is the most common cause of dementia, other neurodegenerative diseases such as Parkinson's disease and Huntington's disease negatively impact lifestyle and family. Therefore, it is vital to discover an effective treatment to slow the progression of neurodegeneration. Rivastigmine is a drug currently used to manage dementia caused by Parkinson's disease and Alzheimer's disease. It prevents the hydrolysis of acetylcholine, a neurotransmitter that is deficient in Alzheimer's disease and Parkinson's disease, by binding to and inhibiting acetylcholinesterase and butyrylcholinesterase. Rivastigmine is especially effective because it also inhibits butyrylcholinesterase, thereby reducing the hydrolysis of acetylcholine more than other cholinesterase inhibitors that only inhibit acetylcholinesterase. However, rivastigmine may have additional uses beyond dementia treatment. The aim of our study is to investigate the effects of rivastigmine on phenotypes of neurodegenerative diseases, specifically Alzheimer's disease and Parkinson's disease, through the use of a C. elegans model. C. elegans is a nematode that can be genetically modified to express certain symptoms of neurodegenerative diseases. Its transparency also enables the utilization of fluorescent proteins to monitor the progression of the disease. With its short lifespan, small genome, and complete nervous system, C. elegans has been an attractive model for neurodegeneration. We studied the transgenic strains CL2355, CL2122, NL5901, and OW13. CL2355 expresses amyloid beta formation in neurons, while CL2122 expresses amyloid beta formation in constitutive muscle. Both are Alzheimer's disease models that are marked with green fluorescent protein, expressing this in the intestines. NL5901 and OW13 are Parkinson's disease models that both express yellow fluorescent protein in muscles containing human alpha-synuclein. We will perform the Nile Red assay on age-synchronized NL5901 nematodes to assess the effect of rivastigmine and analogs on lipid content. We predict that exposure to rivastigmine will lead to increased lipid content in NL5901 compared to the control.
To test this out, we plan to use the Nile Red assay and age synchronization. Age synchronization makes it simpler for us to study C. elegans since it enables us to keep the worms at the same age. This is a crucial step because it removes biases from measurements of the lipid data in each of the worms because lipid composition and aging are associated. Because the brain is filled with lipids, they play a key role in brain function. Normally, when lipid homeostasis is disrupted, neurodegenerative diseases like Alzheimer's and Parkinson's follow. To actually conduct age synchronization, you have to use the bleaching technique, which depends on the fact that while embryos are protected from the bleach by the eggshell, worms are still sensitive to it. Embryos are then incubated in liquid media without nourishment after being treated with alkaline hypochlorite solution, which permits hatching but stops further development. Additionally, the C. elegans was stained in order to measure the amount of lipids. The lipid distribution through tissues can be observed and measured using Nile Red or oil red O stains. When employing the Nile Red stain, C. elegans tissues are examined for a green fluorescence that may be distributed uniformly or in clusters. However, for the oil red O stains, the animal's clear body is contrasted with red lipid droplets. Next slide.
Lipids play an important role in brain function. The brain is enriched in lipids, and a disruption of lipid homeostasis leads to neurodegenerative diseases like Alzheimer's disease. Aging is also known to be associated with lipid composition. Cholinesterase inhibitors represent a significant fraction of currently available drugs as they primarily enhance the cholinergic neurotransmission in the brain by delaying the degradation of acetylcholine available in the synaptic clefts. Rivastigmine is one of these inhibitors that will allow the increase of lipid concentration in C. elegans. We haven't been able to complete the Nile Red staining assay yet, but based on our research and our background knowledge, we predict that our control group will show a normal amount of lipid concentration in lipid bodies when stained. The NL5901 worms that we will use for this assay will be induced with alpha-synuclein after being exposed to a temperature upshift and will show a lower concentration of lipids after being stained. And then the NL5901 worms that have been induced with alpha-synuclein and have been given rivastigmine will show more lipid concentration compared to the NL5901 worms without rivastigmine. Next slide.
To conclude, rivastigmine has been proven effective with dementia, and we believe that with its properties, it can be used to treat neurodegenerative diseases like Alzheimer's and Parkinson's disease. Using the cholinergic hypothesis, which states that the decline in acetylcholine leads to cognitive impairment, we can reason that rivastigmine's inhibition of the breakdown of acetylcholine blocks the enzymes acetylcholinesterase and butyrylcholinesterase, which improves cholinergic depletion. Additionally, since lipids play an important role with many neurodegenerative diseases, using the Nile Red assay, we were hoping to get an insight on how C. elegans strain NL5901 was affected when given rivastigmine. Although we haven't been able to gather conclusive results, we hypothesize that the lipid concentration will be higher in the strain exposed to rivastigmine than the control strain, meaning that the strain would likely be affected by Parkinson's disease. Next slide.
We would like to thank our advisor, Ms. Ranganathan, for her guidance, and we'd also like to thank the lab staff and Mr. New for their advisory. Lastly, we would like to thank ASDRP for giving us this opportunity. Thank you.
And here are references.